speaker. Thank you, Wayne. Our next speaker is Dr. Christy Hagan from the University of Kansas Medical Center. Her talk will focus on PR actions in ER-positive breast cancer.
Thanks, Carol. I'm just waiting for the mic and my slide. Thank you. Thanks, Carol, for the introduction. Thanks to the organizers for allowing me to have 15 minutes to convince both the virtual and live audience why PR is an attractive target in luminal breast cancer. So this audience knows very well that the majority of breast cancers are ER/PR positive upon diagnosis, and as such, we've spent many years devoted to understanding how ER and estrogen promote growth in breast cancer. However, the role for progesterone remains still largely understudied. PR for many years has been tracked as a readout for estrogen receptor. ER is a transcription factor, and it regulates key target genes in breast cancer. One of those key target genes is progesterone receptor itself. So in many tumors, ER and PR expression will track together. And in fact, PR expression is a predictor for how well women will respond to endocrine therapies, as it's an indicator of how well ER is acting in those tumors. That's visible here in these survival curves where survival is shown based on ER or PR expression. And you can see that the highest, that the ER/PR positive tumors have the best survival rates following endocrine therapies, and loss of PR leads to a significant shift in this survival curve. So despite how well ER and PR positive tumors do in response to endocrine therapy, many of these women will progress on their therapies, suggesting that there's more to the picture than just estrogen. So I'm going to spend a moment talking to you about what we know about progesterone in breast cancer. And I want to highlight that all the data I'm going to show you have caveats. None of them were designed perfectly with the question of asking how does progesterone affect breast cancer growth. So please keep those caveats in mind, and many of them are highlighted in some of the reviews that you see referenced at the bottom of my slides. So the first is results from the Women's Health Initiative trial looking at the risks and benefits of hormone replacement therapy. In these women were treated with either estrogen or progestin, which is a synthetic progesterone. Women treated with estrogen plus progestins were at a higher risk of developing breast cancer as compared to women who were treated with estrogen only. These data are mimicked by another trial that was done in the UK where women self-reported their hormone replacement therapy usage. These women who took estrogen plus progestins were at a higher risk of developing breast cancer than estrogen alone. Progestins are synthetic progesterone. I forgot to mention that in the previous slide, and this becomes important in the next slide. In a trial in France, they stratified breast cancer risk based on the type of progestin used in the hormone replacement therapy. And they found that women who were taking progestin, HRT that contains synthetic progestins were the ones that were at higher risk. However, women who were taking HRT with native progesterone did not have an increased risk of breast cancer. So this brings us to kind of my first point, which is that progestins, synthetic progestins, do not equal the actions of native progesterone. So the ligand becomes very important in this situation. Switching now to animal modeling data, this is data from an ACI rat model. These rats, when continuously exposed to estrogen, 100% of these rats will develop mammary gland tumors by 36 weeks. When you add progesterone to this treatment regimen, the development of tumor time is accelerated. And when you block progestin action with an antiprogestin like mifepristone, we see that it almost completely ablates tumor development, suggesting that estrogen and progesterone together are required for the development of mammary tumors. This is in contrast to, however, what we see when we use established tumor models like T47D or MCF7 human breast cancer cell lines, ER/PR positive cell lines. We see in these models that when these cells are grown as xenografts in nude mice, they require estrogen supplementation for growth. Interestingly, in established tumor models, when you combine progesterone, this leads to a decrease in tumor growth, suggesting that in this situation, progesterone is anti-proliferative. This mimics some clinical trial data that was done in the 1970s and 80s, where in many small cohort studies, high-dose progestins were actually used for the treatment of ER/PR positive breast cancers. And in fact, many of these trials were small, but they reported response rates, some up to 45%, in late-stage tumors that were treated with high-dose progestins. So I'm going to pause for a minute and make sure that we're on the same page about 3 basic assumptions that I've tried to cover so far. The first is that not all progestins are created equally. So progesterone does not necessarily equal the actions of synthetic progestins. And that's likely because synthetic progestins can affect other nuclear receptors, some of which we'll talk about in this session. Second, the effects of progesterone likely vary by cancer stage. So in some of those animal modeling data, we're looking at the development of mammary gland tumors, whereas some of those established tumor models or small clinical trials with high-dose progestins, we're looking at established tumors, established cancer. So the effects of progesterone likely will vary by the stage of disease. And finally, more studies are needed in humans. This seems obvious, but I make this point largely because many studies are done in mice, and mice actually are a pretty poor model for luminal breast cancer. They don't develop ER-PR-positive tumors. So unfortunately, humans, as someone said earlier, humans are the best model to study human cancer. So what makes PR the receptor an attractive target in breast cancer? So if we think about the hallmarks of cancer, PR really makes its way into every hallmark of cancer. And if we think about progesterone, sorry, my pointer's not working. If we think about progesterone's role in proliferation as potentially controversial, I presented some proliferative and anti-proliferative data. This highlights that perhaps proliferation isn't the best readout for progesterone action in breast cancer. So I'm going to highlight 2 different vignettes for you, the role of PR and progesterone in stem cells as well as immune evasion, in which the readout may be better for progesterone action in breast cancer. So the first is to highlight work from multiple different labs, many of them listed here over the last 20 years really, studying the role of PR and progesterone and its ability to promote breast tumorigenesis through modulation of either the normal stem cell compartment or the cancer stem cell compartment. This is a schematic here showing a normal duct. Inside the normal duct, PR-positive cells are actually few and far between. The PR-positive cells release secreted factors which act in a paracrine fashion on nearby PR-negative cells. One of those populations of PR-negative cells are mammary stem cells. And in fact, these paracrine actions lead to expansion of this normal stem cell population. This work has then translated into paracrine effects on cancer stem cells. So again, the same theme appears to apply in that PR-positive cells release paracrine factors that release secreted factors that act in a paracrine fashion to promote survival and self-renewal of cancer stem cells. So together, I really like this because this image really highlights the continuum from early stage disease to late disease. If we think about PR and progesterone's roles on either normal or cancer stem cells, those would be most likely viewed in these early stages of disease, so in situ disease, movement to early invasive disease, where survival and stemness may actually be the readout for what PR and progesterone do in this scenario. On the other end of the spectrum then would be proliferation as a readout in established disease, where there is data to suggest that progestins may be anti-proliferative in this setting. So here the readout's proliferation and we're at end stage. So again, highlighting stage of disease and the appropriate readout. The second vignette I'll tell you about is work from my lab and others that looks at the role of PR and progesterone in promoting the tumor microenvironment and how that supports the growth of PR-positive tumors. So early work from my lab really detailed PR transcriptional and multi-step downregulation of the STAT interferon signaling pathway, and we published a number of papers in regard to showing that PR is a potent attenuator of this pathway. Why would this matter clinically? So interferon signaling, shown here is a JAK-STAT signaling pathway that ends in a downstream readout, which is activation of key target genes called interferon-stimulated genes. We showed in those papers I referenced that PR and progesterone can block every step of this pathway. And this translates to be important in early developing tumors where we're understanding that interferon activation is one of the early danger signals that are presented to the surveilling innate immune system. And so you can imagine if this developing tumor, sorry, this pointer just doesn't work. If this early developing tumor is a PR-positive tumor, if it can downregulate those early danger signals to the surveilling immune system, this may allow this tumor to evade immune detection. And recent work from my lab, Lauren Werner, a graduate student in my lab who has a poster tomorrow, has shown that PR and progesterone promote immune cell changes in the mammary gland that support the development of PR-positive tumors. She did this using both a PR transgenic mouse as well as some syngeneic mouse tumor cell lines. So these syngeneic tumor cell lines are great because they allow us to study the isogenic effect of PR-positive versus PR-negative cells in the context of a competent immune system. However, the rate-limiting factor is that almost every syngeneic tumor cell line doesn't express PR and definitely doesn't express ER and PR. So in order to study PR effects in these cell lines, we virally transduce them to express mouse PR. But this gives us an isogenic system to compare non-PR expressing cells to PR expressing cells. Shown here is growth of the 67NR cell. This is one that grows syngeneically in BALB/C mice. The unmodified tumors grow very nicely in their syngeneic hosts. When we transduce them with either the empty vector or the mouse PR, we saw that the empty vector was enough of a xenoantigen to lead to rejection in these mice, and this is commonly seen with different selection markers that are found on vectors. Interestingly, when we express mouse PR, we can now rescue this tumor growth, and these cells can now grow. We know that this PR-mediated rescue of tumor growth is mediated through the immune system, because when we grow these cell lines in skid beige mice lacking fully competent immune systems, we now see that these tumor cell lines grow exactly the same, suggesting that PR's ability to rescue tumor growth here is immune-mediated. To start honing in on the cell types that are important in this immune mediation, we repeated this experiment using CD8 T cell depleting antibodies, and we saw a similar phenotype where in the absence of CD8 T cells, the PR positive and PR negative, or the PR positive and lacking PR cells grow the same, suggesting that CD8 T cells, not surprisingly, are important in this immune-mediated rescue. We know that growth of these tumors is dependent upon PR when we treat them with two different PR antagonists, mifepristone, a classic PR antagonist, and onapristone, a new, more specific PR antagonist. We can completely abrogate tumor growth in these models. Finally, we expanded this into a second syngeneic tumor cell line, EO771s, again, an ER-PR negative cell line that we transduced to express mouse PR. We grow these in their syngeneic hosts, C57 black 6 mice, and in this experiment added exogenous progesterone. And we saw that the addition of exogenous progesterone promoted tumor growth in these mice. And we know that this tumor promotion, this increase in tumor growth is dependent upon the immune system, because again, when we repeat this experiment in skid beige mice, we no longer see that progesterone promotes this tumor growth, indicating that progesterone is promoting tumor growth through an immune-mediated mechanism. We can look at the immune cells in those tumors to try and get an idea of what immune cell changes might be promoting these changes or might be responsible for these changes. We see that progesterone leads to an increase in regulatory T cells, which would be suppressive in the immune microenvironment. Progesterone leads to a downregulation of CDC1s, which are important in tumor antigen presentation. We see a decrease in CD8 positive T cells, as well as a higher level of PD-1 positivity in these T cells, together suggesting that progesterone is promoting an immunosuppressive immune microenvironment. Cumulatively, these data suggest that PR and progesterone can promote immune cell changes that allow for the immune evasion of PR-positive tumors. So my last slide and some final thoughts to leave you with is that proliferation might not be the whole story. We know that ER and estrogen are major drivers of proliferation. PR and progesterone might not be as much. They might be more involved in stem cell maintenance or in what I just presented, protection from the immune system. So we think of ER and estrogen as fuel for the fire, growth for the tumor, and we think of PR and progesterone more as shields from the surveilling immune system. More studies are needed in humans with native progesterone, and there are some of those trials ongoing right now. And there are also some ongoing clinical trials, window of opportunity trials, with two different antiprogestins, telepristone acetate, as well as onapristone, looking to see at what types of changes we see in these early window of opportunity trials. So with that, I will stop there, and I'm happy to take any questions.
Thank you, Christy. There's a couple questions that have come in. A quick one is, what dose of progesterone is critical for tumor growth? Do you have a feel for dose?
Yeah, so that's interesting and something that we've been thinking about. We started with 30 milligrams of progesterone, which sort of mimics high cycling levels of progesterone. And in our experiments that I showed showing that progesterone promotes these proliferative effects, that's the doses that we used. We're discussing right now doing some scale-back trials with lower doses to see if we can achieve the same effect with lower doses, or perhaps we see opposite effects.
Great, thank you. So here's a question from Dr. Nevin again. A secondary metastatic ER-positive, PR-positive breast cancer after long-term estrogen deprivation often becomes PR negative. Do you think that high-dose progestins would work if PR is lacking? Should we be pre-treating tumors with estrogen prior to PR to switch PR back on?
Wow, that's a complex
Complexity.
I'm not sure. I mean, I think you need PR for the correct anti-progestin to work. One of the risks would that be the classic anti-progestins work through other nuclear receptors. Suzanne will talk about GR. That's a classic off-target effect of some of these anti-progestins. So I'd be very careful, and I think that's why these preclinical data that we're doing now in these window of opportunity trials can help us learn more about these next generation PR antagonists that might be more specific to targeting PR.
Great. And a last question I have is, you looked at the CD8 T cells. Did you look at other immune cells, such as infiltrating macrophages, in your studies?
So the data that I presented to you are the changes in cell types that we saw that were significantly changed. We had a large immunoprofiling panel that included macrophages, included natural killer cells, and that's reported in that paper, the entire panel that we investigated, and the data that I showed you here are the immune cell types that were significant.
Thank you so much. I think that we need to try to get back on time. We're still running a bit late, and we need to respect our third speaker. So thank you very much, Christy.